Hello Biotechnicans, this is Dr. Farhan Zamir from Biotechnica, Bangalore. For today, we'll have a great discussion on billion-dollar question, and this billion-dollar question is, will drug discovery using bioinformatics tools ever achieve 1,000 novel molecules per year? Okay, so to understand this particular question, let's try to dive in. Welcome back. So we are now at a juncture of understanding and refuting with a particular question, and this question is, will drug discovery using bioinformatics tools ever achieve 1,000 novel molecules per year? Now, as I need to tell you that with all the tools and techniques, with all the technology, we are simply not able to get 1,000 novel molecules with a greater bioactivity. Either it could be any kind of communicable diseases or on to a non-communicable diseases. Just to tell you, my dear friends, we do not have one precise drug which can work on atopic dermatitis. So, this is the condition. So we cannot boost around saying that we know a lot of chemistry or we know a lot of biology, or now with the advent of bioinformatics, we know everything. So what I would consider as whatever we know can be piled up into just, 1 or 2% of the entire mother nature, what the nature knows. So hence, but the question is, I am trying to put up my constant effort, and when do I reach this particular target of 1,000 novel biomolecules? At least not per month, not per 6 months, at least per year, I should come up, the entire world should come up with at least 1,000 novel molecules. And when this can happen, we can actually, target various kinds of, pathogens, various kinds of viruses, various kinds of fungal infections, and various kinds of bacterial infections. So with this, let me try to, dwell upon before we go into understanding why is it not possible currently. Okay, we need to understand what is the current methodology which has been applied, and before we understand the current methodology, we need to also understand the traditional drug discovery process. Now, let me try to explain you the natural product drug discovery and drug development, especially from herbal products. Okay, so the initial component is you start with a selection of a plant, and then you authenticate the plant and the plant material with the help of a botanist. And then what you do is you start extraction and isolation. Please remember, until now it was the job of a botanist. Okay, from this point onwards, from the extraction and isolation part, okay, here comes the analytical skills. You should know what exactly is the component of polarity, whether your phytochemical is able to miscibilize or polarize with that of your aqueous extract, or again aqueous cold extract, or hot extract, or solvent hot extract or cold extract. So you need to understand the polar and non-polar molecules, that is very importantly phytochemical molecules. And then once you understand this, once you have the extract, then we go for the next step, which is called as physicochemical, understanding the physicochemical characterization and structural elucidation. So once I understand the structural elucidation, we will always go with the biological activity or the biological screening to know whether on a dose dependent manner, or it is called as activity guided assay or bioactivity guided assay, will guide me whether in this particular fractions, whatever I have isolated, which one is an active fraction. So now I would just stick on to that particular active fraction, and then I can, using the power of the technology, especially the instrumentation, I can look into either LCMS, or I can go into GCMS, or I can look for MALDI and then characterize the entire protein or characterize the entire biomolecule. So once I have the entire biomolecule, that is the entire ligand molecule, now with the power of bioinformatics, I can go for molecular docking and simulation studies. So once I do this, since I have isolated it from the plant molecule, it becomes very, very difficult for again going back to the raw material and then extracting it. So hence what we do is we go for a technology which is called as retrosynthesis. Now in retrosynthesis, I am exactly mimicking the natural component, but however this has been done in my chemical laboratory. So this is called as retrosynthesis. So what we do is we go for simply a chemical synthetic method, and once we have the chemical synthetic method, we also need to ensure whether it is having certain toxicity or whether it can absolutely show ADME properties, that is absorption, metabolism, excretion, and the distribution properties, so that I understand that it actually covers ADME and does not show any kind of toxicity. So this is what was the traditional way of drug discovery pipeline took place, either in terms of the chemical component or in terms of a phytochemical component. And then this was then later followed by either in vitro analysis or in vivo analysis. And then the next step was, once it qualifies your preclinical trials, then the next component was the clinical trials. So once you have the clinical trials, then finally the data was been reviewed by FDA. Okay, and once the FDA says that okay, your data is it's trustable, it is reproducible, and then it is rapid. Okay, then the drug is actually released into the market, and once it has been released into the market for a period of around 5 years or so, the drug is always under surveillance. So this is how a traditional drug drug discovery pipeline actually goes on. So to be very specific, we'll try to discuss also on how exactly clinical phases or clinical trials they go on. There are various phases in clinical trials. There are 4 major phases. The first phase is wherein you are looking for the safety and the best dosage level which has to be determined. The second is with the response to the new treatment is also recorded and analyzed. And in third, the results studied from the regulatory systems again are been revised and revisited, and finally it has been approved, and finally at the phase 4 the treatment is being marketed. Okay, so and this has been done with various population of people. So normally they are called as YOPIs, YOPI. Y refers to young, O refers to old, P refers to pregnant, and I refers to immunocompromised patients. So my drug has to qualify all the 4 different situations, and that is how I know that the drug is safe at various kinds of parameters or indices. Now, the process of drug discovery, normally again they are being classified into 3 major types. The first stage is the drug discovery phase, wherein now I take multiple molecules. Here is what I talk about. I can start my research with either 10,000 phytomolecules, or I can start with 10,000 chemical molecules, synthetic molecules, and from there I go on to filtering them at various levels based on either by in silico studies, in vivo studies, or in vitro studies. And that is where your second phase will come in, which is called as preclinical development. So from 10,000 ligand molecules, now I filter down, I narrow it down to 250 compounds. Okay, and once it has been 250 compounds, the next component is the clinical development. So that could be either 5 molecules, and finally the regulatory bodies will have their own intervention, and depending on their approvals, finally you have 1 molecule which gets approved if you are lucky. Now, just because you started with 10,000 molecules, I cannot assure that you will always get 1 lead molecule. But however, if you are lucky, okay, you can still get 1 or 2 lead molecules. This was, the classical method in which a drug development pipeline actually proceeds. But however, now you have the latest trends in drug discovery and development, which has to be implicated, and it has to be understood in a much better way to understand how do I come up with at least 1,000 novel molecules at the end of one particular year. Okay, so for this, I need to have various kinds of strategies. The first strategy is combinational techniques. So rather than going for one single technique, okay, I need to have a combinational approach. And this is where at Biotechnica, a lot of emphasis has been given on to transdisciplinary, interdisciplinary, intradisciplinary. So we need to have an integrated sciences. So, everybody should have a communication with another group of people. So rather than competing with each other, we need to collaborate with each other. When we collaborate with each other, that point science becomes much more easier. So combinational techniques is one of the important key. Then this combinational techniques will lead into high throughput analysis. Now high throughput analysis is also a major norm, because I do not have time to analyze each molecule. So can I design a robotic arm which can do it, do all my analysis at one stretch for like 10,000 molecules or 10,000 ligand molecules could be analyzed at one stretch? If I'm able to do this, then high high throughput sequencing is of great meaning to me. Then very importantly is miniaturization. This is an important component wherein people are trying to look into, and if you have reading news 3 to 4 days back, CEO and managing director of Biocon, she Kiran Majumdar Shah, so she explained that how exactly Biocon can go on to a next level. Okay, very importantly with the trust area of biosimilars, something which is called as biosimilars. So you still have chemical entities which can match biomolecules, which can match proteins in your body, so that rather than creating toxicity, it becomes self in my body, so that the efficacy of my particular drug can actually increase. Now, most of the drugs, many a times they are not able to penetrate or percolate or traverse the blood-brain barrier, and hence all my drugs, many a times the drug actually fails. Now there is a concept wherein if I can miniaturize this particular drug, wherein there is a vehicle which actually transports this particular drug across the blood-brain barrier, okay, then you have a greater efficacy. Now certain people have a different kind of an approach to miniaturize this particular drug molecule, and this approach is a newer technology of biotechnology which is called as nanotechnology. So the implication of nanotechnology, bringing a macromolecule to a micromolecule, from a micromolecule to a nanomolecule, plays a very, very important role. But however, we need to also pay attention that at a nano level, the physical and chemical properties, what you have seen at a macro level, entirely differs with that of the nanoparticle. So hence that's the difference between graphite and graphene. Graphite shows an entirely different properties and graphene shows an entirely different properties. So when I'm converting into its miniature form, the very important component what I need to remember is I need not to meddle with the physico-chemical properties, especially with the toxicological components of it. Then there's something which is called as microdosing. So rather than giving at a larger dose, so I would still make sure that my drug slowly gets released into the host, so that the efficacy is for a very longer time. There's something which is called as the turnover time of a drug. Okay, so this turnover time of a drug could be increased with the decrease in the concentration of the drug. So hence the side effects could be actually minimized. There's another major component which is called as chemometrics. So chemometrics is another component wherein I am actually looking into the chemical component and then rating it in such a way that it could become an efficient drug. So these are the major components which have been actually used, various parameters which have been used for modern pharmaceutical analysis, which can lead into the drug discovery platform. Now these are the various stages. The three major stages is you will identify the hit. Okay, then you have the hit to lead, then you have the lead to optimization, which is called as hit ID, then hit to lead, and then lead optimization. These are the three major steps which have been normally used for identification of a ligand molecule, wherein you can translate a ligand to a drug molecule. Now very importantly, there are various kinds of analytical tools, and also there are various kinds of databases and various kinds of bioinformatic tools which can play a very, very crucial role. So your things could be made easier rather than directly jumping into in vitro studies or in vivo studies. I would always welcome you to take up bioinformatics as one of the major approach, so that you can understand the target well, you can also understand the ligand well. So what you can do is, with all these 10,000 molecules, you create a big data, and with this big data, once there are certain lead molecules which will qualify it, then you can go for in vitro or in vivo studies, or you can go for cell-based studies. And once you do it, then you can also use QSAR, that is quantitative structure activity relationship studies. You can look into the molecular docking component, you can look into the ligand-based pharmacophore analysis, and then finally you can look into the various kinds of similarity searches, that how exactly my particular ligand matches with the other ligand or other drug which is already available in the market. Okay, and how best is my ligand than compared to a drug. So because we started with phytochemistry, so many a times these phytomolecules are simply non-toxic, but however, to increase the efficacy, I need to do certain structural modification, functional group modifications, which we call it as derivatization. Now upon derivatization, if you are able to come up with a very, very potent molecule, that's a eureka moment. So what I urge everybody is, we need more bioinformaticians. India needs more amount of bioinformatics, more quantum of bioinformatics knowledge has to be inculcated into our school and college education, so that our students can think way ahead. Because once they understand the laws of bioinformatics, implicating that into drug discovery becomes much more easier. Because once you have a drug for various kinds of diseases, you can actually bring the economic burden, okay, familial burden, okay, so that the every family or every society can actually have a holistic life. So to create the civil society, the important component is, we will try to understand biology in a much better way. And here we have Biotechnica, which always straves for a better understanding of biology by various kinds of the workshops, by organizing various kinds of certification program, various kinds of internships, so that our students have been empowered, and our youth, when they have been empowered, they are able to think better. So with this particular note, I would say biotechnology and bioinformatics, all put together, all life science put together, do not segregate as life science and computer science, etc. So now I have students from various kinds of different kind of background learning bioinformatics. So when they learn bioinformatics, what they say is they have a complete different kind of paradigm shift in their thinking, and this thinking is necessary for this current scenario. So I welcome everybody to learn bioinformatics, and this is how we can have 1000 novel molecules per year. And if you ask me, sir, is it possible? And the final answer is yes. And this S is only possible with your learning of bioinformatics. Thank you very much for staying connected. So stay tuned and make sure that you subscribe to Biotechnica. Thank you very much. All the very best.